Thank you to Coffee for the invitation to speak today. I don't often get to speak about my research. Mostly I get to front audiences like Robert does to tell them about how great UQ is. And I've got a really polished story there. Today you're going to get a very clunky story, one that is a journey through the last 8 or 10 years of my research and thinking. I've listed myself there, and okay, I'm the person presenting, and throughout this talk I'm going to give you references to the bits and pieces of work that I'm talking about in case you want to follow anything up. But what you will notice is that my name's not on them. It'll say Kapitke et al, it'll say Blamey et al, it'll say Wang et al. I'm in the et als. Okay, so hopefully I can make a legitimate claim that I've been the guy in the background there saying, "Ooh, let's look at this," or "that doesn't make any sense," or "how do we attack this thing? There must be a better way of understanding this." So I'm claiming a whole lot of research here. You'll have to judge for yourselves, and Pax is in the audience, but I don't have to worry about Pung and Peter saying, "Bullshit, he didn't do that," or "You didn't even read the manuscript." That wouldn't be true, but... Okay, so let's have a look at this. Our group have for a long time, so from the time I was doing my PhD, the group of plant nutritionists at UQ have been interested in how metals move out of the soil into plants, and for that matter then how they move on into animals, be that us, be it a grazing animal, be it a koala. Our specific interest as a research group is this one, so out of the soil and into the plant. We don't get involved in research which looks at how bioavailable is metal in a plant once it's eaten by an animal. We work with other groups at UQ, the guys at Entox in particular, who do this really well. So we don't get into that space, but we're in the soil to plant part. And soil scientists have always been in the soil to plant part. We have always been in the how do you predict the availability of phosphorus or nitrogen or potassium or copper or zinc or whatever. So we get some dirt out in the paddock and we bring it home to our lab, and then we'll shake it up with some kind of extractant and measure whatever it is that we're interested in that extractant. And the basis for the thinking here is that there is some predictable relationship between how much metal you might extract from the soil with water or with acid or with Coca-Cola. It doesn't matter. The concept's the same. There's some sort of relationship between this that we can extract and measure and how much would be taken up by the plant. So I want to start this talk by looking at how effectively we can predict the availability of metals that are in the soil, how much is going to get into the plant. So this is a data set, a huge data set actually. Some of this is our own work, so there's little filled in dots there. You won't be able to see very many of them. There's a couple of hundred UQ generated data points, but we also went into the literature. We pulled every paper that we could get data out of to look at. So there's literally thousands of literature sourced data points in here. So we're able to have a really comprehensive look at this, far more than we could do on our own resources or within the one project that funded this work. And so we know that the measure of total anything in a soil is a rather poor predictor of what's going to get out of the soil into a plant. So it's not really a surprise here as we look at total metal content that it's not a very good predictor of getting into the plant. I've also plotted on these a plant toxicity threshold. So this is data for zinc. That's the point at which, so the plant tissue concentration, the point at which the plant starts to get sick because it's got too much zinc. And this one, the animal toxicity threshold, this is the point at which an animal starts to get sick because it's eating plants with that metal content. Does that make sense to you? So in this instance, the plant's going to get sick and grow rather poorly before the animal would get poisoned. That's not the same for all metals. If we're looking at cadmium, it's very much the other way around, so we can poison ourselves or our animals long before the plant starts to feel sick. So that, as far as it goes, that's an absolute fail on its ability to predict metal uptake by a plant. There'd be no point in us measuring total zinc. Hello, Peter. Welcome. And saying, this is how much we'll get into the plant. However, these are the sorts of extractants that are routinely used and routinely assumed to work. The one at the bottom here, DTPA. Actually, DTPA is a very clever extractant. The guys who developed this, Bud Norvell and Willard Lindsay, were thinking about plants which were growing in soils which were deficient in the metal. And so they were looking at how does a plant that's growing under low metal concentrations go about getting as much metal as it needs. And those of you who are into plants and soils and roots know that the plant will produce organic ligands to push out into the soil to complex those metals, and then it'll take them up. So DTPA was to try and mirror that plant behaviour. So it was a really good way of measuring availability of heavy metals, copper, zinc, etc., in low supply conditions. But then people have just taken that and used it at extremely high conditions. And did it work? Well, think about the data that's in the literature. You typically publish the stuff that did work. It's an awful lot easier to publish a paper saying, we got this strong relationship between DTPA and plant uptake than it is to say, well, we got all this rubbish. So by going to the literature, we're imposing a publication bias. We're seeing data where this extractant worked. Okay? Nearly all of the papers we've looked at here, the authors were saying DTPA gave us a good prediction of zinc uptake from these soils. But of course, when we plot them all on the one graph, we just get total rubbish. So a fail in this instance too. In fact, our real problem here is that every soil extractant that's been invented and used fails. When we wrote the paper on this one, there was an extraction method called DGT, which appeared to be giving reasonable predictions, but in the intervening years, as more and more data has been published, if you do the same exercise as this, you get the same result. So a very, very poor prediction. So much as I'm proud to be a soil scientist, we're not very good at what we're supposed to do, are we? And maybe we're biting off more than we can chew. Maybe the task is just too hard here of saying, predict from the soil what we'll get into the plant. Let's try a very, very much easier prediction. Let's say, can we predict what's in the soil solution getting into the plant? So rather than worrying about the solid phase, we can extract soil solution, we can look at what's in the soil solution, we can look at what plants take up. In fact, we don't even need to be in soil solution, we can just do the job in solution culture and look at what a plant takes up. So the same sort of exercise. Peter hit the literature, collected up all of the data sets that he thought were well conducted, and I'll come back to what I mean by well conducted in a minute. So that the papers that were published which had data in them that he thought was acquired in a reasonable manner. Collected up all that data, collected up for all of these metals. What people said was the point at which we saw plant toxicity. And as you can see, these are huge ranges, couple of orders of magnitude there. Now, part of this may be differences between species, but it's not all differences between species. So what I want to do is, in the first instance, look at one metal. I'm going to look at lead and consider why we might get the wrong answers for that and some of our own work. And then I want to look at metals in general and why metals are toxic to plants, and I call it the search for commonalities. So we'll get into the search for commonalities. So the answer you get for what level of lead is toxic is really a question of how you go about doing your experiment, and you find huge ranges. So here's one from one of our papers. In fact, all of our work, we find that lead gets to be toxic to plants at somewhere less than 0.1 micromolar. But if you go and hunt for other papers, and this one by Wang, and there's more recent papers which will tell you, oh no, lead's not toxic until you get to hundreds or even thousands of micromolar. So you see the range difference here. 4 orders of magnitude between when we can kill plants with lead and when other people kill plants with lead. You wouldn't think it'd be that hard, would you? It is. Simple modelling exercise. Put the solution concentrations for our nutrient solutions into a thermodynamic calculation program, so GeoChem or Freak, or there's lots of these things out there. You could do it long-handed, it'd just be a lot of work, so there's no magic in the computer program. So I put 2 solutions up here. Firstly, Hoagland's solution. Bizarrely, people use Hoagland's solution to do plant nutrition work. And I say bizarrely because, have a think about this. We know that in soil solution, in even a very fertile soil, you're going to find maybe 1 micromolar phosphorus. If we're talking about a tropical soil, we're going to be 0.2, 0.5 micromolar phosphorus. And yet Hoagland's solution's got a couple of thousand micromolar phosphorus in it. So it's nothing like what plants are actually experiencing. In fact, when you put plants into Hoagland's solution, they typically have a bit of phosphorus toxicity about them. And certainly any Australian native plant would just die, wouldn't it, Pax? They would not like this at all. So I say bizarrely because, you know, we're trying to understand what plants are experiencing, and you go and hit them with 2,000 times their normal exposure to phosphorus. But it is relatively commonly used, or quarter-strength Hoagland's. The other one here, dilute nutrient solution. This is where we've tried to keep the concentrations of the elements in solution around the levels that you would find in a soil solution. So this is really work that dates right back to Hartley-Teakle. Yes, Hartley-Teakle really was a person. He was a soil scientist. Is that why I'm condemned to Hartley-Teakle building? I don't know. But Hartley-Teakle worked back in the 1950s, and he was able to extract soil solution, and despite the analytical challenges it represented, he was able to measure phosphorus and show that soil solution phosphorus is 1 or 2 micromolar. And so the Australian mentality for plant nutrition, Pax and Colin Asher and the guys here at UQ, Zed Rengel in Western Australia, these guys have all worked with low concentration nutrient solutions. So I want to look at what happens when we put some lead in these solutions. If I can get my machine to talk to me. Okay, so most of our experiments run at pH 5.5 or higher, and here's our lead concentration. So here's the lead in solution, and we get to pH of about, whatever, 3.5, and suddenly lead drops out of solution, and we've got this lead chloropyromorphite, so lead phosphate chloride precipitate. So in that Hoagland's solution, by pH 5.5, okay, it doesn't matter how much lead you started with, it's no longer in solution. In fact, it's quite interesting. If you do the calculations, some of the rates of lead addition are so high that they will have precipitated out nearly all of the phosphorus. So maybe we're seeing phosphorus deficiency limiting plant growth rather than lead toxicity. Now, great experiments. You know, phosphorus supply experiment? No, it was a lead toxicity experiment. So Peter didn't use any of these. He threw all those ones out. The Yang experiment I was showing you, it would have been up here. Okay, so we eliminated all those stupid ones, and yet we've still got these huge ranges down here. Even for us working in dilute nutrient solutions, we still have a real limitation as to how much lead we can get to stay in solution. So here we are. This is plotted a slightly different way. So we're saying that at pH 4.75, we can still only maintain in solution for an indefinite period a concentration of lead less than 0.5 micromolar. So we're still very constrained in what we can operate, the range we can operate in. Remember I said that we'd shown that lead was toxic at 0.1. So this really isn't an experimental constraint, and nor is it a real-world constraint. Okay, if lead couldn't be held in solution above 0.5 and it was toxic at 3, then lead toxicity wouldn't exist, would it? It would not be a problem in the environment, and lead toxicity is a problem in the environment for us. So I wanted to track through some of our experimental work. A lot of our experiments are conducted in a manner similar to this. So a dilute nutrient solution, a pH that's slightly acid, so it's easier to keep our heavy metals in solution, but not acid enough that the plants are suffering from any sort of a H+ challenge. Okay, it's not that acid. We use 22-litre drums, renew the solution if we're running for any extended period of time. So in this particular experiment, multiple rates of lead, but it would normally be multiple rates of whatever metal that we're interested in. And we typically have included Rhodes grass in our experimentation. Rhodes grass is the gold standard tough grass. So if you want something to plant on a mine site to get, stabilisation of the landscape, and it's saline, and it's dry, and it's challenging in all respects, Rhodes grass is a reasonable start, provided you've got enough rainfall to support it. The other grass was signal grass, and the sort of data we generate, and I won't dwell on this for very long, in this instance showing decrease in yield as we increase the concentration of lead, and Rhodes grass performing a lot worse, okay, same scale at the bottom, so performing a lot worse than the signal grass. So signal grass is actually tougher than Rhodes grass. But the point I wanted to make with this, and so here we've shifted to, okay, same concentrations of lead on this axis, but here we're looking at the concentration of lead in the shoots. And if we put that same threshold for an animal diet across there, it rather changes what you might think about signal grass. So where we might have been thinking, oh gee, signal grass would be great, we can grow that on lead contaminated tailings, that's going to be brilliant. But actually what you end up doing is transferring the lead out of the tailings into the signal grass, which is going to be growing perfectly happily. You're still at where are we? About 50% yield there. 50% yield of material, which is more than twice what it would be good for an animal to be eating. So if we did this, if we rehabilitated a tailings dam with signal grass and we used it then for grazing because it all looks great, the grass is growing fine, we would have this problem of transfer of metal to the animal and to the animal that eats the animal that would be us, and we should all be not happy about that. We spend a lot of time looking at roots. If you want to join our research group, have a passion for long hours spent looking at roots. Love microscopes. So here's the sort of, and here's why we look at the roots, because it's the roots you can see the impact of the metals. The impact of the metal is on the root. Before you see any damage anywhere else, in fact, looking at the tops and the plant's not doing well, that's often for heavy metals because the roots have been damaged, and that's what's stopping the plant getting its nutrients, getting its water, growing well. So, happy, healthy roots here, so a happy, healthy root tip. Further back from the tip, root hairs growing out, all, fine, beautiful, healthy specimens. As we increase our exposure to lead, so here's 0.5 micromolar, and we've got tip damage and a bit of swelling and discoloration here. By 1.1 micromolar, we've got absolute huge damage and no further growth of the root tip. And then here at 3.5 micromolar, the root tip's shot, but also the root hairs are a real mess. So by looking at the roots, we get this real easy insight into, is this metal damaging our plant? And we can do this really quickly. We can grow young seedlings, grow them in solution, pull them out, stain them under the microscope. What have we got here? Signal grass, different concentrations. Remember before I was going 0.11, so 10 micromolar, so 0 happy roots, and at 10, still pretty happy roots. I mean, there's a bit of damage here, not too bad. And interestingly, there's all of this white precipitate. Can you see that? That there's all of this white stuff on the roots. And when you stain this, so we used a stain which shows lead, This is lead precipitate. So we're thinking, this is really cool, protection mechanism. This plant has worked out some really neat way of precipitating the lead out outside its roots. Okay, because remember, signal grass is much more tolerant of the metal than the Rhodes grass was. The Rhodes grass didn't have that white stuff around its roots, it just got sick and died. And here we've got signal grass with pretty happy roots and heaps of precipitate around them. Tolerance mechanism, wonderful stuff. And then we thought, oh shit, let's pause for a minute. Remember precipitation. Remember this graph? Huh? Precipitated here. Where's 10? Out here somewhere? So of course what we were doing was running a solution to get to levels high enough that the signal grass was damaged. We were way outside that level at which we could sustain the metal in solution. And even the slow kinetics of precipitation weren't protecting us. The metal was precipitating out, and just because the roots are a little bit stickier than the sides of the beaker, the metal precipitate all accumulated on the roots. How close was that to embarrassment? I'm pretty sure Peter would have had the draft manuscript on his computer ready to go, wouldn't he, Pax? He gets into these things so fast. So fortunately we paused and thought for long enough. And so even for groups who are, as it were, doing the right thing, it's so easy working with heavy metals to make mistakes which, yeah, you can then publish and regret at your leisure. That's an error, but let's move on to some of the things that did work for us and some of the truths revealed as opposed to the embarrassing artefacts that we managed to generate. Here's the standard model of what happens when a heavy metal enters a plant root. If you look up any standard textbook, this is what it's going to tell you. So here's our lead going into our cell, and the plant responds by producing metallothioneins. So these are organic ligands which are capable of binding to the metal and detoxifying it. We absolutely know this happens, so every textbook tells us that this happens. We know it happens. I call it the gene jockey's view of the world, because you can see this by looking at the genes that are upregulated by the plant's biochemistry. So there's no question that this happens. We don't do plant biochemistry. We're not that clever. And so it left us free to see other things. And when we looked, we did see other things. So here's our signal grassroots, transmission electron microscopy. The dark patches that you see here are lead precipitated out. So remember, the metallothionein thing is the lead goes into the cell produces a ligand to bind the lead, and it holds it in solution bound. Okay, detoxifies it, but it holds it in solution. And that's not what we're seeing here, is it? We're seeing lead as solid phase, X-ray impermeable precipitate. So what's going on? What is this stuff? So here's a couple of reference materials, so possible precipitates, the lead phosphate chloride, so chloroporomorphite and a lead phosphate. So either of these is possible. Here's our picture of the deposits, and that little circle represents the point at which we are taking the analysis of what is in that precipitate. And there's lots of lovely toys on transmission electron microscopes that allow you to probe what are things made out of and what's their X-ray pattern, etc. And so you can see for both of these, as we've probed what its structure is using energy dispersive X-ray analysis, we see the little chloride peaks there. So very likely to be chloroporomorphite, and in fact if we run repeatedly, so 10 or 12 reps, 10 reps on the same thing, we get this nice little chloride peak. That's the dark line, the full line is the plant lead, the dotted line is chloroporomorphite, and the bottom line is the phosphate. So what the plant is clearly doing here is precipitating out the lead as chloroporomorphite. And you might say, well, why would it do that? That's the most insoluble form of lead there is. Okay? So this is rather a clever strategy by the plant. The plant thinks, oh, there's some lead coming to me, this is going to be bad, I'll precipitate that as chloroporomorphite, being a bit anthropomorphic there. But so the plant's getting rid of the lead as a precipitate. Rather than spending a lot of photosynthate, building a lot of metallothionein, and instead of having a whole lot of this stuff floating around inside its cell, it precipitates the lead out. Even more interesting is what it does after it precipitates it out. So here's our lead precipitate, and what we see over time is that gets shifted out of the cytoplasm, out into the cell wall, okay, out into the, as it were, dead part, out of the living cell. So once again, isn't this a clever strategy? Precipitate the stuff out, move it out of the cell. We think the Golgi apparatus has a role in that only because we see these precipitates associated with the Golgi apparatus in a lot of our images. And then, of course, you only have a limited amount of lead left in the cell that your metallothionein would have to deal with. So it's a way of getting rid of the vast bulk of the contaminant it's got into. Let's move on. The search for commonalities. So this is, let's not just look at lead, let's look at a whole suite of metals and see if there is any common behaviour. Is there a standard way in which metals all make plants unhappy? Short-term experiments, so this is Peter's lab bench. This is where he conducts most of his experiments, and Pax and Pung, they've got great light set up in there, so exposing the plants to enough radiation to grow happily. But the short-term growth means that the plant's not actually dependent on that photosynthesis. There's enough nutrient in the seed, and there's even enough energy in there to grow for a day or so. So, you know, the photosynthesis is a small contribution, but especially the nutrient in the seed means that the plant doesn't really need to take up nutrients. Most of our experiments we conduct using a complete but dilute nutrient solution. Sometimes we can't do that. So if the metal that we want to look at has a very insoluble sulfate or a very insoluble phosphate, we'd use a simpler nutrient solution. So sometimes the nutrient solution is as simple as some calcium chloride and some boron, so that we can get a, you know, the roots need calcium and boron all the time in order to grow in a healthy manner. So we typically use a full nutrient solution, but not always. Short-term growth, and our measurement is of root elongation. So both how long the root has grown is our measure of growth, but also then looking at the roots, looking at what kind of damage is on them. As I say, we spend lots and lots of time looking at roots. Pax must spend hundreds of hours in each experiment because there'll be multiple roots per rep, multiple reps, multiple rates, and then multiple metals. So here we have metals: aluminium, copper, lanthanum, gadolinium, indium, all sorts of things. So metals, not necessarily metals of interest to us because they're environmental toxins. We just went through the periodic table, literally saying what metals are going to help us to understand what could be driving the toxicity. We were looking for particular types of characteristics in the metals, not necessarily that they were of environmental importance. So we look at the roots and look at hundreds of pictures, and Pax is saying, you know, I've seen 50 that look like this, but only 30 that look like that. Do you think that means anything? And, gee, there's a lot of them that are twisted. Do you think that means anything? What about this? Do you reckon that means anything? Because you can see there's every root's different, and what's coincidental here and what is meaningful. That's what we have to tease out. We are more and more interested in ruptures. So by rupture, what I'm talking about, if you look at this root up here, you can see that's the outside of the root there and there and there. But then this is a break. There's sort of, you can see right down into the workings of the root. Something has gone wrong here and the root's torn apart. So we became very interested in those ruptures. And we started to ask, do all metals cause rupturing like that? And this is a more difficult question than it might seem at first. Remember that we're often dealing with metals that we know nothing about. So first of all, you've got to work out at what sort of concentration does this become toxic. There's no point in us looking at it at a concentration that's so toxic the plant's just dead, doesn't grow at all. And there's no point in us looking at concentrations that are a couple of orders too low to cause toxicity. So first of all, we had to do a lot of messing about to get in the right range where we might limit the plant's growth so that the root's intoxicated by metal, but it's not completely killed by the metal. So we're seeing this point at which the impact of the metal is first apparent. Oh, and let me go back there. And what we find is a set of metals that do cause ruptures and another set of metals that do not cause ruptures. So now we're saying, wow, is there a reason why there's these two groupings? Is this able to tell us something about the mechanism by which these different metals might be toxic or not? So, the ones that cause ruptures, here's our attempt at a rationale for what might be going on there. So we're saying that the metals bind to the cells on the outer part of the root, bind to the walls of the cells in the rhizodermis and outer cortex. And that this increases the rigidity of the cell. Now remember how a root grows. So at the tip of the root you've got division of cells, more cells being produced, but then behind the tip you've got a zone of elongation where the cells get much, much bigger. So it's not actually the division forming new cells that's really growing the root, it's the zone of elongation where little cells become very much bigger cells. And what has to happen there is that the cell has to allow the cell wall to expand. Can you see that? Yeah. So somehow the cell wall has to be allowed to expand. And we're saying by the metals binding onto it, somehow that process is prevented. The cell wall can't expand. And so we've got the inner part of the root that's protected from the metals, growing at its normal rate, and we've got the outside where you've got this constraint of cell walls not able to elongate, and the root splits apart. That's our hypothesis. And so it's a hypothesis based on observation. We've got no mechanism here yet, have we? We're just saying, this might be what's happening. We really need a chemical mechanism for what could be happening that would cause some of these metals to prevent elongation and others not to prevent elongation. And when we plot them all in this way, so here we're plotting a hard ligand scale. So hard ligands are things like carbonyl groups and phosphoryl groups. Carbonyls in particular we see in cell wall pectins. Okay, so yep, those are things that we would see in the cell wall that these metals up here would be strongly attracted to. And then these metals down here are the ones that don't form ruptures, that are sitting low down on this hard ligand scale. So it appears that this hard ligand scale might help us to predict what metals are going to cause ruptures and which are not. So once again, we're pretty content with ourselves by this point, and would have remained content, I guess, except Pax said, oh, what about silver? You know, silver occupies an interesting place in this spectrum. What about silver? It shouldn't form ruptures, should it? Because it's way down there on the hard ligand scale. Unfortunately, when Pax went about looking at this, damn, perfectly good theory, shot to pieces on the basis of one metal. Silver forms beautiful ruptures outside our theory. But silver's a rather unusual metal. It's way more toxic than those other metals down the bottom there. You know, they were things like copper and potassium and, you know, metals that we wouldn't consider to be very toxic on that hard ligand scale. But we know that silver is extremely toxic. And so we're left with, we have to content ourselves with the view that silver's causing ruptures, but probably by a different mechanism. It is very high up on that soft ligand scale, so it's binding to other parts of the cell wall extremely strongly. So it may be that it's causing the ruptures in the same way by preventing elongation of the cell wall, but it's not doing it by binding to the same ligands that copper or mercury might be binding to. When I did my PhD, one of the real breakthroughs of the time, so work that Pax did, was the recognition that if you went from measuring concentration in solution to activity, and for those of you who aren't chemists, concentration is how much metal is present per unit volume of solution, whereas activity is a measure of the capacity of that metal to participate in a reaction. So we say in an infinitely dilute solution, whatever that means, the concentration and activity are the same, but as the solution becomes more concentrated, the activity drops relative to the concentration. So it's a measure of how easily the metal can participate in a reaction. And when Pax plotted data for aluminium using activity rather than concentration, he was able to show very, very much better correspondence between the plant response and his activity measure. So we've taken one step further now. We've said, gee, the cell wall is charged, and so is the plasma membrane. They're negatively charged. So they're going to attract cations, metals, out of solution into the cell wall. So the concentration within the cell wall, so the concentration right beside the plasma membrane, is going to be very different to the concentration outside. Does this make sense to you? So you've got a charge, a negative charge there, pulls the positive cations in, so concentrates them up. So looking in the box solution isn't actually telling us what's at the plasma membrane, where the plant's going to be affected by it. So Pung's a brilliant mathematician. I'm not even going to bother to tell you that he's a brilliant soil scientist, because all soil scientists are brilliant. But he's both a good soil scientist and a good mathematician. So he can calculate these things for us. What's the effect of the cell wall charge? What's the effect of the plasma membrane charge? What will the activity be adjacent to the plasma membrane? And so here we've got the same data set, so different concentrations of calcium in solution and a range of copper. So okay, at any particular copper concentration, these dots represent different calcium concentrations. What the calcium is causing is the charge on the cell wall to differ. Okay, so looking at the box solution, we get a rather poor prediction of copper toxicity. A lot of variability here in our data. Is it very, very toxic, or is it not very toxic at all at 0.5? It depends on how much calcium was around. We used to think this was the calcium somehow protecting the plant from the copper. But when you look at what's at the plasma membrane, you get this one. So check the change in concentration, or change in activity rather. So this is ranging up to 2, 2.5. This is ranging up to 200. So there's a 10-fold concentration from outside to inside the root. So obviously we do need to take that into consideration in our thinking. But when we do, we get this much, much more accurate prediction of what level of the metal is going to be toxic. And taking this same game to the extreme, so there are lots of tricks we can use to change the charge on the cell wall and on the plasma membrane. We can change the ionic strength of the solution. We can change its pH. We can change how much aluminium is in there, because aluminium will adsorb, changing the charge. So by adding aluminium to a level that's not toxic, but as much as we can relative to zero, by messing around with the pH, raising the pH and lowering it, but not raising it so far that we precipitate our copper out, we can get this very wide range of charge on the cell wall. And when we do that, we get very, very, very poor prediction of toxicity, so root elongation, relative to how much copper is in solution. But when we express these, once again, calculated as to what's at the plasma membrane, we get much better prediction. So in other words, we've stepped one step closer to understanding what is going on, and one step closer to actually being able to make a prediction, even on the basis of measuring the simple solution. Remember, I started by wanting to make a prediction on the basis of what we could extract out of the soil? You can see how many steps we are on from that. It makes me more and more pessimistic that any extraction from the soil is going to give you a useful prediction of toxicity to plants. So, my last shot at the common mechanism of toxicity. Here's our data sets for 26 metals. Toxic metals. Now, think about this. I've got things like copper, in there. Yep, that's a toxic metal. But I've also got things like calcium and potassium. Yeah? So I'm considering these all toxic. If you give a plant enough potassium, you will eventually elicit a toxic response. And if we find potassium in here, you can't see the scale, but that's a log scale, so 10,000, 100,000, a million. So we're talking very, very concentrated solutions relative to something like, let's find copper, where, you know, this is 1 here. So 1, 10,000, huge differences in the concentrations or the activities that we need to see that toxic response. We're trying to find the point at which we're getting about 50% yield reduction. And then we're taking that value and we're saying, against some measure of chemistry, where do we see a relationship? So these things like Pauling electronegativity, the radius of the hydrated ion, these are the sorts of things chemists look at to say, how can this ion participate in a reaction? These are all things that will control or influence the chemistry, the chemical reactions. And most of them, yeah, no sort of prediction at all. It's only these ones at the bottom, and if we click forward, the hard ligand scale, which I showed you earlier, it's the only one that's giving us a sensible prediction of the point at which the thing becomes toxic. So once again, I would say we're stepping one step at a time closer to a point where we can say, gee, we think that we have an understanding of what might be going on here. Let me stress that we're talking about one part of the toxicity of the metal to the plant. This is what you see happening in the very short term. It's controlled by the metal binding onto the outside of the roots. It's not controlled by the metal being taken up, redistributed through the plant, doing all sorts of other things, which some of these metals do. So this is just the first stage. It's just one part of the metal toxicity to the plant. And you'd wonder why we'd spend 10 years to understand one part of that we have. Okay, so here's our logic. Here's what we think we understand at this point. These metals which bind strongly to the hard ligands bind to them in the cell walls. Remember, cell walls are made up of a whole lot of fibrils of cellulose and hemicellulose in a pectin matrix. And in order for the thing to elongate, the pectin holds it all together. You've got to get the pectin to release so those fibrils can slide against each other. The way that the cell does that, the pectin's got a lot of calcium in it. When it's gelled, the cell pumps some H+, so it acidifies the cell wall. The H+ displaces the calcium. The pectin releases the fibrils. They can elongate. And then the cell raises the pH of the cell wall and they lock off again. Beautiful. Very, very simple. When the heavy metals get in there, the H+ can't displace those metals. So by acidifying the cell wall, the plant still can't get those fibrils to relax and slide against each other. So that prevents the cell wall from elongating. So we've stepped from an observation of we see ruptures to now mechanism. This is how the metal could be preventing elongation and causing the rupture to form. And I had to throw this in, so I hope I've still got a couple of minutes to speak. Robert hasn't told me to shut up yet. The synchrotron. This is fun stuff. Those of you who have not played games on the Australian synchrotron, exercise your mind. Find some way that this fits into your research and get down there. What a fantastic scientist boy's toy. $1 billion piece of kit. Wonderful, wonderful. And here's Bridget loading samples in. In our labs in Hartley Teakle building, there's a photo of me loading samples in, and they say that's just before Neal broke the machine. And there's an element of truth in that. So they don't invite me back as often as they used to. We've done most of our work using X-ray absorption spectroscopy and X-ray fluorescence. For absorption, we need to freeze our samples in liquid nitrogen, but for fluorescence, we can put them in as hydrated, ambient temperature samples, so literally a happy, healthy root. We can look at it with the synchrotron. And the beauty of the synchrotron is that we can see the bonding arrangements for the metals that are in there. So what I wanted to show you is that we can use the synchrotron to see bonding arrangements. To this point I've been saying, because of the chemistry, we think that copper should be binding to the pectins in the cell wall. Okay, that's on the basis of chemistry. The synchrotron should allow us to see the copper bonded to the pectins, if our theory is right. I'd also have to say here, Peter is a brilliant guy because he's worked out limitations to the Australian synchrotron such that he has to go and use the synchrotron in Italy. You know, the dedication to science. I mean. Okay, here's the sort of data that you get out. So at the very bottom here we've got lines for our roots, 3-hour roots and 24-hour roots. And then above this we've got lines for a whole lot of standards. So the approach here is that you compare what you see in your sample to standards or to a group of standards, and it's a curve fitting exercise. What of these standards describes the sample that we see? I've chosen zinc and copper here. Zinc is one of the elements that wasn't forming ruptures. Copper is one of the elements that is forming ruptures. So our theory is the zinc does not bind strongly to the cell wall pectins and hence doesn't prevent elongation, whereas the copper does bind strongly to the cell wall pectins and will cause ruptures or prevent elongation, cause ruptures. So when we look at where the metal is here, we should see zinc not bound to cell wall pectins and copper bound to cell wall pectins. So if I just step you through this, so down at the bottom here, our 3 and 24-hour roots. So this is to see whether there's a time effect. We ran roots that had only been exposed to metal for 3 hours and ones that had been exposed for 24 hours. And I know it's a bit of a look up and down, but of course the computer software does this for us. So we can tell that that phytic acid at the top there, that's what the zinc in these roots is bound to. And there's lots of other work that using different techniques that tells us when a plant's got some zinc, it will complex it with phytic acid, it'll move it to the vacuole, it'll store it there until it needs it. So this is a plant dealing with a plant nutrient. It's not trying to protect itself from it, it's really storing it up to use later. And of course by binding it to the phytic acid, it's making sure that there's not excessive levels of free zinc floating around, which could be problematic to it. So this is not a cell wall component. So far we're doing all right. Okay, the zinc is exactly where we'd expect it to be, or not where we don't want it to be. It's not bound to the cell wall component. At 3 hours, our copper is bound to cysteine, which was a bit of a surprise. Cysteine is one of these sorts of metallothionein type compounds, a protection compound. So at 3 hours, the plant's coping with how much copper's coming in by complexing it, leaving it in solution. But by 24 hours, we've got a lot of the copper up here. So this is polygalacturonic acid. This is the pectin in the cell wall. So once again, this is what we were predicting. We're saying copper prevents elongation because it binds to the pectins, and the synchrotron could see that. And of course, some lovely photos. As a soil chemist, I go along to conferences and everyone's got great photos of stuff, and I'm a chemist, I've got boring stuff like tables and graphs. So it's a real treat for a chemist to be able to say, look, here's some chemistry pictures. These are pictures from the synchrotron, our work using the X-ray fluorescence line. These are our two-dimensional images of roots, and these ones are tomography, so these are computer-generated from two-dimensional scans. What we've done here is, you can see the green line, this is a little capillary tube. We've put a live root in the capillary tube, it's full of water. We've put that in the machine, and we've been able to blast it with super high intensity X-rays and measure the fluorescence coming back off it to see what metals are in there, what their speciation is. In this instance, we're just looking at where the metals are. And so if we look at metals like nickel, The nickel's largely concentrated inside the root. Zinc the same, and you can see some zinc in the steel there, so going into the phloem, heading up into the plant. And that's also fits with our prediction. These were metals which don't bind to the cell wall components, so they're going into the cell, into the root. Whereas copper, and even more so mercury, are bound to the outside of the root. So here's mercury. You can see the accumulation of mercury right around the outside of the root. Very, very little getting in. Whereas zinc, yeah, there's some around the outside here. There's some, stored in vacuoles, and there's some being moved to the xylem to go up through the plant to the tops. Nickel. So nickel is a plant nutrient, but remember, plants need very, very little nickel. So we're seeing it mostly stored in vacuoles in the cells, and we can't see any, there's no accumulation in the xylem, so the plant isn't exporting much of it to the top. So this is a very consistent story of the pictures telling us, showing us pretty much what we would expect to see. And even showing us over here with the copper, the formation of the ruptures that we'd expect. The root expanding out and breaking away, and the copper being concentrated on the outside bits of the root that are left behind. So, to conclude, I think we are at a point where we're satisfied that the chemical binding to hard ligands, the binding to the cell wall components, explains why these particular set of metals that form ruptures do so, and why the metals that don't form ruptures don't. Peng's gone on to develop better models for us, models that can separate out the osmotic effects. Okay, remember we said we had to get calcium and potassium to huge levels in order to create a true toxic effect, but there's an osmotic effect, there's a salinity effect there as well. So Peng's able to separate out those multiple components of salinity, of toxicity, so that we can even refine our predictions somewhat more here. But to return to where I started, does this help us in any way to predict the soil to plant thing? I'd have to say not very much. Okay, we now understand what form the metal will be in the solution, how it has an impact on the plants. We increasingly understand where it might be stored in the roots and whether it's sent to the tops. And of course that's important. We really do want to know, is the toxic metal going to get to the tops and be a problem to us? And of course, turn that on its head. We're nearly all copper and zinc deficient. We're nearly all copper deficient. We're marginal for zinc. So, Robert, you're involved in this sort of stuff, trying to get more heavy metals into grains like rice. Understanding how the plant deals with the metal in its root is also part of that pathway of what can we do to shift metals that we want to build up in the tissue into the tissue. Metals like copper, as you could see, bind strongly to the cell wall components, and it's pretty hard for the plant to take much into the tops. Okay, I'm happy to field questions.